Hey Genetics Gang, I'm Mahek,
I'm Tara.
and we want to tell you about selective breeding and how it affects the greatest meal of the year.
Thanksgiving dinner.
Let's start with some sides.
Our first delicious and selectively bred food is corn.
Selective breeding is when two members of the same species are bred to get the desired characteristics. Corn started out as a small, hard-to-eat plant called
Teosine.
which European settlers and subsequent farmers tweaked to make into the starchier, less colorful, sweet corn that we eat today by breeding corn that have the sugary 1 and shrunken 2 genes together. This makes the corn more delicious to eat.
Americans have only ever eaten one type of turkey.
The broad-breasted white turkey.
However, back in the early 1900s, turkeys were eaten straight from the wild with minimal consistency between the birds. When the demand for smaller turkeys to fit into ovens and fridges arose, selective breeding was utilized between quick-growing birds with
bigger breasts and white feathers were selected
to produce
broad-breasted white turkeys.
Smaller birds are being preferred today, with the macrochromosome number 27 is one of the factors that controls the size of turkeys that we enjoy by determining its height.
Pumpkins were selectively bred to get bigger pumpkins with better flavor. The pumpkins that had the best flavor were bred with each other to produce more pumpkins with the more desired taste. There are many genes that affect the flavor of pumpkins, including
the 79 loci gene and the 48 polymorphic allozyme loci gene.
The pumpkins that were bigger were also selectively bred together to produce the maximum size pumpkin because the smaller ones weren't preferred. The pumpkins that were bigger were selectively bred together to produce the maximum size pumpkins rather than smaller ones by breeding the pumpkins that had the big size alleles, since there are many alleles that control the size of pumpkins.
Now that you know your food isn't exactly natural,
Happy Thanksgiving! Okay.